Thanks, Darren. So, Chris, some of you already know what I'm doing, but we've been looking at water communities, surface water deserts throughout the Trans-Pecos. We're kind of looking at some disease stuff with bighorn sheep and the like. If y'all don't already know, bighorn have a disease problem. They have an pneumonia problem in general. There's been dozens of epizootics throughout the western half of the country. Basically, nearly every population outside in the U.S., outside of Alaska, has had some sort of epizootic. Big die-offs, causing lots of problems trying to keep these populations in high numbers. Previously, Texas, we kind of thought we were safe. Typically associated with domestic sheep, we don't really have a lot of domestic sheep in Trans-Pecos, and so we thought we were good. Well, that bubble was burst in 2019 in the Bay of Horns, and 2 more times since then with Blackout in 2020, and then Diablos in 2023. Like I mentioned, we always thought everyone believed it was domestic sheep was a problem since they've been brought over hundreds of years ago. They've seen more and more problems with these livestock giving diseases to wildlife. Domestic sheep are typically the culprit again with transmitting pneumonia. But again, thought we were safe, and it turns out the issue we think now is ODAT. You've seen the areas are everywhere. Numbers anywhere in the Trans-Pecos, 10,000, 50,000. We don't know. They're just covering all over area, causing some of the habitat problems and everything else. So they are capable of clearing it. They can die, but it is exceedingly rare for them to die. More often, they have it, they'll transmit it to others, and then they clear it and they go on with it. So the history of what caused pneumonia has kind of been figuring it out for about 100 years now, if not longer. It was initially believed 100 years ago it was a lungworm. They kind of ruled that one out after a while when they saw lungworms in lots of healthy individuals never had any problems, so that's something else entirely. We have now what we call the bighorn sheep pneumonia complex with 4 different bacterial species. Those top 3, Yersinia, Pasteurella, and Hemia. At some point in the last 30, 40 years, everyone thought that was the main culprit, that was the thing causing it. Did a lot of work to try and figure out what it was. It wasn't until about 2008, Tom Besser with Washington State had a paper and kind of figured out, okay, it's actually, we think it's Mycoplasma pneumonia or moping. That seems to be the one that triggers the initial infection. After that, any of those other 3 can get involved, and those symptoms go from here to over the top. That's when you start seeing really, really big problems. You can kind of see the graph is just kind of what distribution was 200 years ago. They really did cover the entire western half of the U.S., and that dwindled over time to lots of different things: overhunting, habitat loss, and then disease and predation. And when we have these pneumonia outbreaks, we can see anywhere from very low initial die-offs. There's been a few strains where it's only about 10, 15%, all the way up to 90% die-offs with some of these more severe strains. The problem isn't just the initial die-off either, though. A lot of the times we have what we call chronic carriers. It's mostly more pronounced the effects are with when it's a youth, but there is a 5 to 20% chance that any individual that gets the disease can end up becoming a chronic carrier. What that is that individual has the disease, won't die from it, but won't clear it. And so it becomes a real big issue in the spring months when you have a group of ewes, the rams separate after all that, the ewes have their lambs, they form little nursery groups. That one chronic carrier ewe, after a couple months, all of a sudden, all the lambs die. They lose whatever immunity they had from their mother after about 2 months, and then they get pneumonia, they're wiped out. So you can see years of 0 recruitment with new lambs because of that one chronic carrier ewe. There's been a lot of work, like what do we do about it from across the U.S. And right now, if you get one and you can ID it, you have to remove it. It's the most effective way to actually save that population and try and restore recruitment. Talked about different strains. Initially, the belief was, okay, if they get pneumonia once, that population should be good. They're finding there's so many different strains that being immune to one strain can offer immunity to another. And there have been multiple times where one population gets hit, not very hard with the strain of pneumonia, like, oh, we should be good now. And then a couple years later, a different strain came in and really wiped out the entire population. So typically, before early on, it was believed it was a nose-to-nose sort of transmission. One domestic sheep had to go up and bump or check out a group of bighorn, Audette check out bighorn, and vice versa, and that was how that was transferred. There's more and more evidence now that it's not just nose to nose. We're looking at water. There's a few reasons for that. There's what are these waters doing, but there's also been evidence just a couple of years ago. Again, Washington, Montana State did some work with Tom Besser and Princess Kasier, and they had separate pins. And you can see on that graph that I pulled from their paper, those top pins had a certain strain. And those top pins that had disease in it were 30 meters from those bottom sets of pins. Those bottom sets of pins ended up getting sick with the strain from the top set. Over 30 meters, there was no interaction of individuals over that 30 meters. And they came to the conclusion that they can be short distance airborne transmission. I usually equate it to kind of the COVID thing. We had a little bit of separation. There was no worry, like if you're outside, you're sitting on the end of the block, someone 2 blocks away sneezes, you're not going to get COVID because they sneeze that far. It is more of a droplet transmission, but there is evidence that it can actually move through the air a little bit further than we initially believed. I kind of learned a little bit of this paper, just kind of doing some homework, but also looking for more water stuff because there's always talk of, oh, so-and-so did some work with water and put Moby in water, gave it to sheep, and got sick. That's kind of how I got in touch with Dr. Besser and this paper where they actually had an instance of detecting it in water, but were not able to regrow it, so it seemed to not be viable. In addition to that, also that same year, y'all know Logan Thomas was with A&M, he's now at Kansas State. They did some work where they had domestic sheep on one side of the pen and they had bighorn on the other side with the tarp in the middle, and they shared a water source. And they had transmission. They don't know if it was short distance through the air or in the water. They were able to kind of have that same result that they had in this paper right here. So again, waters, surface waters in the Trans Pecos. Most of you have probably seen it, but it's the northern part of the Chihuahuan Desert, very arid. There used to be a lot more springs than there are today. A lot of the agricultural stuff that was done, a lot of the farming and everything kind of lowered that water table. A lot of the springs that used to run all the time and are now ephemeral or just don't run at all. Because of that, we do it every year with Work Project, but guzzlers are the common conservation solution. Finding a way to put water out in the landscape for animals to use during those droughts. They supply the water fluids, you see them all over the place. One of the other concerns, even if we don't find disease in the waters, is that they become localized sources for animals to interact, and it becomes a way for disease to be transmitted. So with all my work, we have basically looked at 3 different things. The first off is just taking these water samples. What are the bacteria that are in these individual guzzlers? Some of them are really clean, some of them you go out there, they are just covered with algae and everything else. And with learning what those communities are, can we identify some variables that might explain, well, this guzzler over here is growing in this community of bacteria, but this one over here has completely different. What is different about them, and can we find some things that will predict what is growing here but not here, and then go find a random guzzler and see, oh, can we guess what's going to be in there based on these other variables around it. Second objective we're looking at is, can Moby survive in fresh water? So mycoplasmas do not have a cell wall. Common theory is without a cell wall that goes into fresh water, that water's going to rush in, it's going to lyse those cells, that cell becomes dead. That shouldn't be a problem. So we're looking at how long, if you put it in water, how long before it's dead, and we don't have to worry about the water serving as a transmission source. And finally, a little bit different from the water, but still with the Moby, is are there epigenetic differences between bighorn sheep and aoudad that are the reasons, maybe potentially, on why aoudad are far more resistant to Moby and pneumonia than bighorn sheep are. So we're looking at the water sampling for some of this microbiome stuff we're doing. Kind of give you an idea of what we're using. It's just a little 60 ml syringe, a filter, and then the data card that we're filling it out. We're taking the name of the location, so if it's a landowner who's doing stuff for me, they can write on if it's a guzzler, or if it's a stock tank, anything like that. When we're on the WMAs, all the guzzlers have names. It makes it easier on me for data management to make sure I have the right GPS coordinates, because you go to the same guzzler, that guzzler didn't move. It's the same one that helps me with data management. Date and time, because we're looking at seasonality, how much water was actually filtered. Those filters if there's a lot of dirt or algae or anything in those filters, it gets really hard to push water through very quickly. So knowing how much we're able to get through can help explain potentially what we actually see when we get those results. on the microbiomes. And then who collected it, and then if I'm out there, we have a little water probe that we're getting some water quality data with. So we have switched since last time I talked to y'all from 0.45 micrometer filters to 0.22. We went out to Montana State last June, we kind of found out that the 0.45s weren't going to give us the results we needed. So we since pivoted to 0.22, and thus we've had to push less water through those filters because they get very bound up very quickly. We're doing 2 filters per site. I'm going to give a reason on that as we go a bit further. But as we look at sample scheduling, we're trying to get out about 6 times a year. So once in the middle of winter, trying to see what's going on. Theory and a lot of data suggests that sheep don't really use water those times of year. It doesn't get hot enough for them to actively need to seek it out, but they still get it here and there. Once you get in lambing season, try to get out sometime in February and April, and then those kind of peak months when they're most likely to actively seek that surface water would be twice in the summer, so we're kind of going sometime in May as it starts to heat up, and middle of July as it gets real hot, and then I guess what we call the monsoon season is August and October. We try to get back out there. Sheep typically, like I said, those hotter months is when they're more likely to seek surface water. If it's cool, if there's good vegetation, they don't need surface water to survive. They can get it through metabolically through their food, but in that time, when it gets really hot, 100 degrees out, we pull up to a guzzler, and a lot of times we'll see sheep leaving those sites. So the 3 areas I'm going to with routinely are the 3 WMAs in the Trans-Pecos. That is Black Gap, which is down there in the bottom right in your big bends. Elephant Mountain, the one there in the middle. Haley just talked to us about that one. And then Sierra Diablos is there north of Bitcoin. There are 45 sites that can pretty easily get to in Black Gap within a car. The other ones were probably more work. There's 6 on Elephant that I can get to, and then there's 10 in the Diablos that typically run problems with. Anything else requires some of the Adopt a Guzzler programs. People go out in the spring, early spring, late winter, and do some work, and they're getting samples from you then. Other times, if they're flying for herd assessments or surveys, and they kind of stop it off to check guzzlers, we can grab samples in. Also, 13 other samples from landowners, which I appreciate everyone's help on that. And all told, since I started this, we have 814 samples from 400 different locations and times. Look at them individually, so that's Black Gap. Since August 2024, when we switched to the 0.22 filters, we've gotten 223 different samples. Again, 45 locations. There's a little over 60 total, so there's another about 20 or so that have to be flown to, and have the opportunity to get 5 of those in April that are final herd assessments. So we're trying to get some of those little more remote locations that would have required either you're not hiking to it at all, or it's an all-day affair. Elephant, we've gotten 26 samples since 24. Again, 6 locations with 3 others that require a hike or a helicopter trip. And there's 1 spring that I know where it is, but I've actually yet to see it flow. I don't know if it actually ever does, or if it just kind of stays moving around, but it is there. I just not been able to get water from it yet. And probably the Diablos, 15 samples since August. 10 of those I can get to pretty easy. Most of them can be done in the pickup. They require a short hike. Do it in one morning and done. The other 6 is a whole other fiasco. It usually requires helicopter. There's big hikes, and also with Sierra Diablos being totally surrounded by landowners or special permissions required to get out there. 2 times I've gone with Josh, use it as an escort to get out there and get some of those samples. So Diablos are a little bit better working with to get out there. Like I mentioned, the water quality stuff, we're using this probe. It's going to give us some extra data. We think some of these water quality data will kind of fill in some of those blanks on the microbiomes we see. What we're looking at is the water temperature, the conductivity, or some of you see it as total dissolved solids, dissolved oxygen, nitrates, and pH. We've been using that since this May of 2024. Kind of some overviews on some of the numbers we've seen so far is all 3 of those. The ones I noted that are important are the ones that are really in red. There's some data. There's not a lot of data on what's safe for a lot of wildlife species, but there has been work done on cows that saying you can go over 9.5 is kind of when you're starting to cause problems. There's some work out of the Sonoran Desert where they have pH as high as 12, and the authors were kind of like, well, that's not great, but we think actually most wildlife are kind of resilient to those higher pHs. Nothing else is really of concern. I think our even our maximum nitrates of the 72 they're black at was still maybe a tenth of what is actually considered dangerous for animals to drink from. With the total dissolved solids, it's 20%, so it's still well below these thresholds of what could be dangerous for animals. So as far as the health of the water itself, doesn't seem to be a huge issue so far with the water quality we're seeing. It's more about what is this doing to the microfibers. So as we collect those specimens, we're getting ready this fall to start doing some DNA extractions, and we're going to send these off for gene sequencing. What that does is we're looking at the 16S gene in bacteria. That is the gene that codes for a protein that basically builds all of the proteins for that cell. It's really unique for each species, and it's a very well-proven way to get at what species are in each water sample and how. So previously with some of that pilot data that Rachel did several years ago, they were using what's called, it's an older technique called Illumina. It's called a short read. It's really good for finding, if you only have lots of smaller fragments of DNA, you can kind of run through those and give you an idea what maybe you're going in there. But when you say short reads, it's only looking at a couple hundred base pairs. So if y'all remember from science class, there's 4 different bases that basically code for all the DNA in your body, A, C, T, and G. And so only getting a couple hundred of those, it's great if you only have those, like I said, those small sections of DNA. But for some of these more rare species that have not been studied, and some of those algae, those have been studied so much, they know those DNA strands forward, backwards, inside out. So when you send these off to get sequenced, those species come back with a very high probability. Like we are almost 100%, we are basically 100% certain that this species of algae is growing in, or this one. But when you get to some of these less studied, less fully sequenced diseases like Moby, you're far less confident with that small little snippet that that's actually what's there. It might say we think it's there, but you're going to end up with a very low probability of that actually being right. So what we're transitioning to is newer technology. It's probably about 2 years old now. Is PacBio, Nanopore, both have them, but they're called long read. They will look at thousands of base pairs in the sequence, and it's far more effective on those rare species to actually give you a good confidence number to tell you this. We actually do feel good about saying this is Moby in this water sample. This is Beaverstenia in this water sample. It will give us a lot better results to work with and give us, hopefully, a better idea if there's any diseases going on in these waters. So again, as microbiome analysis, the big picture is what do the communities look like? Within that, we're trying to see, toppling, is Moby in there? Do we think that those waters are serving as a transition source themselves for Moby into ammonia in bighorn sheep? Or is it more likely that it hits the water and dies, and you're more likely to transition around those water sources rather than through the water? The next level is, are there any other members of the SPC? Are we going to see some of those others in there? We don't know that. That's what we're looking for. And then third, are there any other diseases at all that we're going to identify as we go forward with this? And not even just bighorn diseases. Mule deer, javelina, all these animals use these waters. How likely are diseases moving because of those guzzlers? Again, can we find any correlate? Like I mentioned, those independent variables we're trying to identify. Is there a way we can correlate that to those guzzlers and see if we can predict what might be going on in any given guzzler? Some of those independent variables we're looking at, kind of try to divide this up a little bit. Those top green ones, that's all the stuff we're doing with mapping. So we can look at the elevation and how steep it is, stuff like that. Northness and eastness comes from aspect, what way that mountain is kind of facing, or that slope is facing. If you use just aspect, the way the numbers work, switch to like even radians where it's degrees. When you just go to run that in your stats program, it thinks 1 and 360 degrees are completely different numbers on polar opposites, when in reality they're right next to each other. So the way around that is you have to turn that into 2 different numbers of how north it face and how east it face in order to figure out what that actual slope is. The vector ruggedness measure is a measure of how rough. It's a metric within GIS packages. Tells you how rugged that area is. And then to escape terrain is considered anything that is a VRM over than 2 with more than half a hectare. And that is considered what bighorn like to have nearby any of these water sources, so they have something to move to. If they see a predator, if they get spooked, they seem to feel more comfortable if they have the kind of escape terrain nearby to run to. geographic range between what area we're looking at, the nearest neighbor, how close. We would think, potentially, the closer two guzzlers are to each other, the more likely those microbiomes are going to be related. That may not be the case, that's what we're looking at. Like the kind of cover type we're looking at. We have the weather stuff. There hasn't rained recently, hasn't snowed recently. UV index we think can be important. UV kills everything, it breaks down everything. It's going to break down DNA faster if it's exposed to heavy UV. So is that going to have an effect on some of the things we see? What was the daily temperature, max and minimum on that given day? Did that have an effect in addition to that water temperature we're taking, as well as seasonality? Like I said, we're recording the day we're there, the year we're there, we incorporate that. Looking at again the water quality data, and that last one was narrow use. So I mentioned we're getting 2 samples. Part of the reason we're getting 2 samples is if we have an issue with that first one, we extract DNA, something goes wrong, we're not able to get that good data from it, we can do it again with the second one. The other thing we want to look at is taking some of images that are of game cameras that are already on site that can tell us when animals were there recently. We can use what's called eDNA, which is typically used, has been used a lot in tracking invasives in water systems and outdoor, and even in the air. You can grab a water sample, you can take an air sample, and you can test for DNA, and it will tell you what's been in the air recently. It's been used a lot in Texas lakes for zebra mussels. They don't have to go and actually look for the actual mussels. They can just take a water sample and find zebra mussel DNA, and they go, oh, this is here. So we want to see how well we can find other animals that have been using that water source through eDNA. How recently can we detect a bighorn used this guzzler 3 days ago? Can we tell if it happened when you used it, things like that? So as we move into the second part of our objective is that water experiment I talked about. We want to know, can Moby live in water, and if it does, how long does it live? So I went to Montana State and we worked with Dr. Ivanka Boyich and Patricia Brewster at Montana State. There we ran 4 different experiments during the 2 weeks I was there of putting active Moby in water and then taking samples out of it and testing to see if it was, first off, detectable. We didn't know if this was going to be an issue where it was going to break down so quickly that I get all the samples out in the wild I want and we never be able to find it. So we want to know its detectability. Detectability is not saying whether it's alive. All it's going to tell us is if that little strand of DNA is still present in the water. It hasn't completely broken down to make it unidentifiable. The other part we'll look at is viability. Viability is whether or not it's still alive. Did it immediately lie, it's dead, or if we pull that back out and put it back in some culture broth, mix it up and let it grow, excuse me, if we let it grow again, will it grow back? Is it still viable? Is there a chance that if a sick aoudad, sick sheep, went and drank from that water, left Moby behind from some snot, it leaves, a day later, a healthy bighorn comes and drinks from it. Can it pull in still living bacteria that would propagate and cause infection and spread it that way? So we were able to do all of this at Montana State. So we spent the last 2 years getting really good at regrowing Moby in culture and then being able to use it. So that was kind of good there. How that worked is we had 5 beakers of water that we filled with 200 mils of water, and then first one was a control. You can see on with the thermometer, you keep track of how warm our water was, the big red label, that was control. All we did was take some of broth with no bacteria in it, put it in there, and just pull samples to make sure, okay, we can't grow anything if there's nothing in the water to begin with. The other 4 were different varying levels of dilution of what we grew. So how we would do this, a couple days before we were ready to run the experiment, is we'll get a bunch of little tubes and put broth in it. I'll show the picture in just a minute. But we would put, one would be the control, and then the other 3 or 4, we would put a little bit of Moby in it, cap it, put it on the incubator, and let it grow. After about 2 or 3 days, it gets a color change, it tells us it's active and growing, and then we can start. We would pull those 4 together, so we had one consistent concentration of Moby in that broth, and then we would build that out. We would put 1 mil of what we would call stock, or 1-to-1 ratio, in 200 mils of water, and that became our 1-to-1 ratio. The other 3 we do what's called serial dilutions. So we do a times 10 dilution on one, that was our times 10. You do a times 10 on that one, which gives you 100, and then once more for 1,000. Once we were ready, we inoculated everything, and we would start the clock. When I got there, everyone agreed. No cell wall, this stuff's gonna die immediately, but we'll see how long we can at least detect it. We'll pull a couple cultures just to make sure that, yeah, it's not growing. Everyone was on board. this isn't going to be an issue. So we pulled first sample at 30 seconds. We did that for both detectability with the PCR that we're going to talk about in a minute, as well as reculturing to see if it would regrow. We then pulled samples at 2 minutes, 5, 10, all the way to 30 minutes. And that 30-minute sample, we also pulled for detectability, and then again cultured that one because surely we'll do that in 30 minutes. You can see there at the bottom, we then shifted up to 72 hours, and we'll talk about that or why. But overall, we ended up with 45 total specimens that we tested for detections and 18 that we used for viability. So you can see in the picture, that rightmost tube, the red, that is what it's called sugar phosphate 4, which is really good at regrowing mycoplasmas. That's what it looks like initially. There's a color change that happens from a pH change. Whenever that bacteria starts growing, it changes the pH of the broth and turns into yellow. That's how we knew it would grow initially for us to start. And that was going to be the color change if we ended up seeing regrowing after a bit of water. So doing this at Montana State, they already have the whole system in place they have really good markers for being able to detect through PCR, and then we had a great place that we could regrow all this. They've been doing it for a while. They work with Working Dogs for Conservation, who is able to do some of the detections, app captures on swabs to kind of tell you ahead of time that they think your sheep have pneumonia. So we were able to get quick turnaround, and we were able to alter this experiment on the fly as we were going to get results. So between the 4 that we did, we got grass for 3 of them. The first 2 we did with molecular water. There is nothing else in this water. There shouldn't be any bacteria. There should be nothing else. So it was going to give us the highest chance of making sure that our PCR reactions would work, and we were able to get it. The way PCR works is it is looking for that string of DNA that's going to tell it eventually. It's called polymerase chain reactions. We're stacking on little pieces until it gets to that final piece that's going to light up and say, yes, we found it. You will get false positives. Usually that takes a long time, which is why we have that threshold. That dotted line across the middle is 26. It's called cycle thresholds. That's the number of times it took to try before it finally got a positive. A lot of times you will eventually have something randomly attached, and so anything above that is considered negative. At Montana State, they're very conservative with that positive number. So they took especially that 1 to 10 line, that blue line. If we were to send those same samples to, say, Washington State, they would probably consider that positive because they don't have that threshold needed to when they're working with Working Dogs for Conservation. Those dogs need a little bit higher concentrations to train on. So as you can see, there wasn't a whole lot of change over time for the most part, but we did see a very large change with what our dilutions were. The stronger our dilutions were, the lower that cycle threshold was, the more positive it was. For our 3rd and 4th experiments, we was just using tap water. We want to see if that changed anything. As far as PCR worked, we didn't really have an issue. Our 1 to 1 was strongly positive, even 1 to 10, 1 to 100, all showed positive. I didn't have the 1 to 1,000 on here. When we ran it never showed. It was the same as the control. There was very low reaction. It was so diluted out, there wasn't enough bacteria left, enough DNA for it to cause any sort of to fly positive on the PCR. So efficient detectability, I mentioned the 1 to 1,000, we stopped using it. This did tell us that as we're out there, as we're doing the sequencing, as I'm out in the WMAs, as we're getting these water samples from the wild, we should be able to detect it with the sequencing. The DNA, if it's not too diluted out, we should be able to find it, which helps us, which is promising in the fact that if it's there, we should be able to know it's there. Again, not with one is, so we're wondering if maybe the reason we were able to find it at 1,000 is because it was too diluted out. Talking to Patty, who we were running it with, is we found out some of those, I think it was the 1 to 100, 1 to 10, 100 is a far more common of what they see from maple swabs. So we think as we extrapolate that out, if you over dilute it, it's not going to be there anymore. And we think that's kind of maybe a parallel for what's called minimum effective dosage. It's the number of viruses, number of virons or bacteria cells that's required to get you sick. Very rarely will one cell of an infectious agent actually cause disease. It usually takes a certain amount to get over the hump of an immune system and everything else to cause that. When I go back, you can see all those, the green G's are where it regrew. So like I said, we switched to 72 hours because at 30 minutes, we found out it was still growing. That was a big head scratching. We don't know what's going on. We were able to reculture those, and after a couple of days, they started turning yellow again and it started showing activity. So we don't know why. Again, everyone we talk to, all of the common sense said, no, it shouldn't live. So we don't know why. So if we don't know, is it just a far greater concentration than we would see in the wild, and there's just a little bit enough left that it can regrow again? We're not sure. What we did do while we were there is took made an extra sample up in the little safety cabinets we were using. They all have an intense UV light that you can keep on to help sterilize the area. So one night we set up an experiment. We took one of those extra vials and went in, we put it in that unused cabinet, closed the door, turned the UV light on, and we let it cook for 12 hours. When we came back the next day, we pulled samples from it. We were able to detect DNA, so the DNA was still there, but we could not get that to regrow. So UV light is killing it, which is great. So we're curious because, again, everyone said work with Besser, work with Walsh. They were never able to actually get anything out of an outdoor sample to regrow. They were never able to cause disease between sheep with ammonia and water. So we think part of the reason could be UV lights. So we are planning to rerun this whole experiment this summer in Lubbock. I should have been 3 weeks into that. We've been working with them to try and get all this done. We had an issue that came up right before we were ready to ship with a transfer agreement that Montana State's Biosafety Committee needs. We've been working on that for the last couple of weeks. I was told before I came here that everything seems good to go. We should actually get some shipping information soon. Get it to Lubbock. We're going to start running this outdoors. We can run on this, sit outside and see what that changes with it. A little bit of a blessing. Usually by this time in Lubbock, it's kind of like as a transparency. It's well over 100 degrees. It's super dry. It didn't do that so far this year. It's actually been pretty rainy in Lubbock. It's just now starting to warm up, so we're kind of hoping we can get those similar weather events going on to kind of really get those intense UV days, very hot, kind of see how this affects it. So we're just going to run it a bunch of times just outside of Lubbock in the parking lot. We built those cages to kind of make sure that birds and stuff aren't going to get into the water, and we don't have to worry about spraying OB into Lubbock, because that would be a whole other issue. We run this a whole bunch. We're going to take some of those samples again for detection. We're going to ship them back up to Montana State. For Patty to run for, we're going to get those solutions back. And what's going to eventually come down to is to get confirmed if it's growing or not. We're going to have to use it with flow cytometry, just fully plate it and confirm this is specifically moving. We're confident that's what was growing, but before to finalize it, we're going to do a little bit extra. And the last thing I want to talk about, a little bit again, shifting gears, less water but still pneumonia, is DNA methylation. We always explain this. So if in one cell in your body, if you were to take all of the DNA in that one cell and you were out to run it end to end, it would be 2 meters long. It's very, very thin, but it is extremely long. And so one of the ways the body and cells are able to compact that is called DNA methylation. It's a normal process that occurs. It really does, it changes with age, as it is a way of storing DNA, but also a way of controlling what genes are in use. So when you're very, very little and you need lots of growing proteins that activate, making you taller, making teeth, all this kind of stuff, there's a lot of proteins that are active. As you get older, the way the DNA reacts is it starts binding up that little section of DNA. It's a mechanism that makes it less likely for the genes in that area to actually be translated and for those proteins to actually exist. What has recently become known is diseases can cause this in people. There's been some work, one of them, there's an E. coli strain that they started doing testing on people that were infected with E. coli, and they found the E. coli itself caused methylation of genes that would otherwise create proteins that would help fight that infection. So basically one of the reasons E. coli could stay around longer is because it was preventing the immune system from even properly responding. So our question has become, what would happen is this a potential reason that oddad are less susceptible and less at risk than bighorn are? Is methylation in oddad, when it's been attacked by pneumonia, does it respond differently? And so we have hundreds of samples of oddad tissues, from captures, from culls, all this kind of stuff. So what we're going to do is kind of similar to the DNA extractions we're doing from the microbiome, we're doing with this. We'll sequence it so we know what it looks like. We'll run a comparison to see, okay, how does oddad methylation change over time? What does the little oddad do? How does it compare to the big oddad methylation? So we want to see how methylation works with oddad. Then we can look at, okay, so we do have some tissues from Audad who actually had pneumonia at the time. How does that methylation compare to healthy Audad? Is there a methylation change that we can detect? For a while when this was first coming out, we believed you really had to have tissue from the infected area, so we'd have to get lung tissue. More recently, there's been some work done with elk that found they could detect methylation changes from hoof disease in elk, nowhere near the hoof. So we're hoping that we can actually potentially find some methylation changes in ears, even though this is a lung disease. We want to see, carry that over if we're able to, or if we're able to, if not, whatever, if someone else is able to have enough bighorn samples, can they find different changes in methylation in bighorn that are affected with smoking? And what's the comparison of that with Audad? Is there something that could be done to kind of help with that methylation issue in bighorn that could help them fight it a little bit better? So that'll be the fun one. And with that, I'll take any questions. Yes, sir.
Ever any evidence of zoonotic transmission between Audads and you?
Audad's and
humans?
no, there's anything I've been able to find there is no risk of mycoplasma of pneumonia affecting humans. So it's the same genus, different species that cause it. So in humans it's mycoplasma pneumoniae. In sheep it's the species is ova pneumoniae. In pigs it's pneumoniae, and there's a lot of different mammals kind of have their own species. There does not seem to be a lot of cross compatibility with some of those. So
What do you do in your spare time?
double check.
Thank you, Chris. That's all we have for our research update. Any more burning questions about fecal samples or PCR? You can catch these two out and about and talk. Tonight, the social hour starts at 5, and then dinner and breakfast starts at 7. Thank you. And tonight it'll be at the big room, that freestanding room, where we are usually for the big, not in here where we were last time.
Are we showing the movie?
Next page. Are we showing the movie today?
I was going to show it. We can show it now. We can show it at the thing. We can do whatever we like.